The Medusa Disease, Turning to Stone, by Sarah Chalmer, Alexandra and Catherine Copley. In the 17th century, a French physician described a case in which a woman turned into wood, which was the process of other tissues turning into bone. This disease came to be known as fibrodysplasia ossificans progressiva, or FOP, which is characterized by the irreversible transformation of soft tissue into bone at extraskeletal sites by a process known as heterotopic ossification, or HO. In FOP, this heterotopic ossification typically follows a pattern of localization, from upper to lower and proximal to distal. This gradually restricts movement and decreases quality of life over time. FOP is universally fatal, with an average life expectancy of around 40 years, although symptoms don't typically present until adolescence. FOP is a rare disease, affecting approximately 1 in 2 million individuals. It is caused by an autosomal dominant mutation in the ACVR1 gene, which stands for activin receptor type 1. ACVR1 is a gene that encodes for a transmembrane kinase receptor called activin receptor-like kinase 2, or ALK2. ALK2 binds activin, a member of the TGF-beta cytokine superfamily, and bone morphogenetic proteins, or BMPs, a group of cytokines that play a role in bone formation. The most commonly identified mutation in patients with FOP is known as R206H, which is a heterozygous single nucleotide adenine to guanine transversion. This leads to a change in amino acid residue 206 from an evolutionarily conserved arginine to a histidine residue. This is a missense mutation in the ACVR1 gene on chromosome 2. FOP has no known sex-specific, racial, or regional risk factors. It is primarily characterized by congenital malformations of the big toes and progressive flare-ups of heterotopic ossification, as well as swelling. New bone tissue will form at the site of any trauma, including minor muscle strains, ligament sprains, bone breaks, epidermal cuts, bruising, or even vaccine injection sites. The symptoms associated with these features include joint stiffness, chronic pain, and growth defects. Individuals affected by FOP are typically wheelchair-bound by the time they reach maturity. The end stages of FOP gradually lead to what is known as thoracic insufficiency syndrome, which is the inability of the thorax, or chest, to support normal respiration and lung growth. This thoracic insufficiency prevents patients with FOP from breathing, leading to cardiorespiratory failure and death. There is currently no effective treatment for FOP. Diagnosis of FOP is performed by a clinical assessment and later by genetic confirmation to identify the particular disease-causing mutation. Osteogenesis. Bone stem cells, known as osteoprogenitor cells, are found in the bone marrow. Upregulation of genes favoring bone formation leads to the differentiation of these osteoprogenitors to become osteoblasts. These cells then secrete a cartilaginous matrix, which eventually becomes mineralized to form calcified bone. The ACVR1 receptor is composed of 4 subunits of type 1 and type 2 transmembrane kinase receptors. Once either BMP or activin A dimers bind the kinase subunits, they tetramerize to form the complete ACVR1 receptor. A competitive inhibitor, 12 kilodalton FK506 binding protein, or FKBP12, competes with the BMP and activin dimers for position within the ACVR1 binding site. SMADs are a set of transcription factors which regulate the BMP pathway. When BMP or activin bind the receptor, they phosphorylate the receptor-regulated SMADs, or rSMADs, which are SMAD1, 5, and 8. Upon phosphorylation, the SMADs translocate into the nucleus to upregulate the expression of genes involved in osteogenesis. In the context of FOP, the mutation discussed previously, R206H, causes a disruption in this pathway. The arginine to histidine residue shift in the receptor causes a decrease in the affinity to the competitive inhibitor, as well as an increased affinity to activin binding. This is a result of a loss of specificity in the receptor type. Because of this, activin will outcompete both FKBP12 and BMP for position in the receptor binding site. This leads to an upregulation in the pathway, favoring the formation of new bone. A recent study by Lee Shepard and colleagues, published in Nature Communications in 2018, used a mouse model of the ACVR1 mutation to examine the role of fibroadipogenic progenitors, or FAPs, in driving the pathogenesis of FOP. FAPs are a resident multipotent stem cell population found in the skeletal muscle. The researchers tested whether FAPs were the source of heterotopic ossification by pinching the hind limb muscles of the mice. They found that it produced new bone tissue 100% of the time in mice with mutated FAPs, whereas the pinch test injury model was not sufficient to induce ossification in mice that had the mutation in other cell types. They next introduced exogenous activin A into isolated cell cultures from the mutated mice, and found that mutant forms of FAPs were responsive to activin A dimers, while wild-type FAPs were not responsive to activin A, but were responsive to BMP2 stimulation. Furthermore, they found in mice that the presence of activin ligands was necessary to induce heterotopic ossification, and that the inhibition of activin blocked heterotopic ossification from occurring in both injury-induced and activin-stimulated experiments. As such, the results of this study suggest that therapeutic intervention for FOP would be most beneficial if targeted specifically towards activin activity in fibroadipogenic progenitor cells. Future research in this area should be focused on early diagnosis and on identifying non-invasive ways to block heterotopic ossification via targeted gene therapy, as well as activin and ACVR1 inhibition. Beyond this, identifying the potential for reversal of heterotopic ossification could prove crucial to improving quality of life for patients with FOP. Despite the fact that FOP is a very specialized disease, the mechanisms involved in its pathogenesis are also involved in several other disease models. In particular, the BMP-SMAD pathway and activin signaling are implicated in two different fatal brain tumors, namely glioblastoma multiforme, or GBM, and diffuse intrinsic pontine glioma, or DIPG. Because of this, figuring out ways to manipulate these pathways could help establish potential treatment options for several devastating diseases affecting a much broader population.